Good day, this is Professor Will, CBMD PhD, and today's Wednesday, April 6, 2022. It is 5:25 and 19 seconds, a.m. Eastern Standard Time. Today's research, DNA discovery. So let's hop to it here. DNA discovery reveals a critical accordion effect for switching off genes. Date April 5th, 2022. Source, Walter and Eliza, Hall Institute. Researchers have revealed how an accordion effect is critical to switching off genes in a study that transforms the fundamentals of what we know about gene silencing. The finding expands our understanding of how we switch genes on and off to make the different cell types in our bodies as we develop in the womb. It also offers a new way to potentially harness gene silencing in the future to treat or reverse the progression of a broad range of diseases, including cancer, congenital, and infectious diseases. The DNA that makes up our genetic material is wrapped tightly around proteins, like thread wraps around a spool. When it is loosely packaged, the genes can be switched on. When it is tightly compacted, genes are switched off. In the new study, the researchers found that to switch a gene off, the DNA packaging must initially loosen up, before then being tightly compressed. Researchers said discovering the accordion-style trigger took the team by surprise, changing their fundamental understanding to date of this critical process. Researchers were amazed to learn that the DNA first needs to relax to trigger this process, and similar to how an accordion needs to open up before it is compressed to elicit a musical note. Researchers found that DNA needs to be opened up first before it can be compressed and the gene is silenced. Researchers said gene silencing had amazing therapeutic potential. If we can learn how to exactly switch genes off, we may one day be able to switch off dimensional genes in a variety of diseases. And if you can switch off the oncogenes that drive cancer, for example, you potentially could have a new treatment. And to be able to realize this dream, researchers first need to know how the process happens so it can be mimicked with medicines. And their discovery is one more vital piece of the puzzle. The fundamental mechanistic study was focused on efficiently searching for new factors involved in the gene silencing process. And to enable this, the team created a system they called XMETs, based on red and green tags that are normally switched off during development. The system reported gene activity from each X chromosome to the expression of red and green fluorescent protein to reveal if the gene silencing process was occurring normally. The study uncovered a new molecular mechanism of gene silencing, but the researchers pinpointed the protein complex required for this process, known as the PAF complex. The next steps for the research will investigate why the accordion effect is required for gene silencing and the relevance of the process for genes on other chromosomes, such as the autosomes. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.